Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome to the next lecture in the third unit of the Plant Biotechnology course. In this unit we are learning about the protoplast isolations, its in vitro culture, protoplast fusion to generate the somatic hybrids and its application for the crop improvement. And today I will discuss about the somaclonal variations and its applications. My name is Manoj Sharma and I am working as an assistant professor of plant biology at Jawaharlal Nehru University, New Delhi. In JNU, I teach plant biotechnology and genetic engineering courses at School of Biotechnology. These lectures have been reviewed by Professor Kashmir Singh, who is working as a professor of plant biology at Department of Biotechnology, Punjab University, Chandigarh. And this project has been sponsored by DTH Swayam Prabha, MHRD, New Delhi. So in this lecture, I will talk about the somaclonal variations, sources of these variations, then possible mechanisms that causes or that result in these variations, and finally I will talk about various methods to generate the somaclones and their applications in the agriculture and crop improvement. The genetic changes or the variations that are observed in the plants regenerated through the in vitro cultures of somatic cells are called as somatic variations. Though these variations have been observed in the in vitro cultures for a long time, but these were usually ignored, thinking these are abnormalities and are not useful. However, in early 1970s, their potential for the agronomic improvement was realized, and since then, lot of progress have been made to understand the biology of the somaclonal variations and their commercial use for agronomic betterment. The term somaclones was coined for the first time by Larkin and Scowcroft in 1981, where soma means the vegetative and clone means the identical copies. That is, the variations which arise in the vegetative clones of the plants are called as the somaclonal variations. Other terms have also been used to describe such variations like the gametoclones, caliclones, or protoclones, or mericlones. These names have been derived from the sources or the origin of these clones. Like the clones that are derived from the culture of gametic cells, that is the microspore culture or anther culture, these are called as the gametoclones. And the genetic variations observed in these plants regenerated from microspores or anthers are called as the gametoclonal variations. Similarly, caliclones is the term given for the callus cultures. Protoclones is the term given for the protoplast culture, and mericlone is for the meristem culture. And the variations observed in these cultures are respectively called as the caliclonal variations, protoclonal variation, or the mericlonal variations. Depending upon the objective of the experiment, somaclonal variations may be desirable or undesirable. For example, if the objective of the in vitro culture is the clonal multiplication of the elite cultivar, like what we earlier studied in the micropropagation, these variations are undesirable, as the introduction of these variations in the elite cultivar can result in the loss of the quality or the quantity of the commercial product, or it may introduce other unintended characteristics. On the other hand, these variations can be an important source for the genetic variability and can be used to either improve the existing crops or to develop the new desirable or novel genotypes that may have agronomic and commercial advantages. These variations may be caused by several factors and can be temporary or permanent, like the changes in the tissue culture raised plants that result from the physiological or epigenetic effects usually do not inherit to the next generation and are reversible, and therefore these variations or these phenotypes which results from the epigenetic changes or the physiological changes, these are temporary ones. Since their identification in 1970s, somaclonal variations have been identified for several traits in plants. Some of the major traits includes disease resistance, resistance to the abiotic stress factors like salt stress or heat tolerance or tolerance to the freezing temperatures. Then several other traits related to the health of the plant or the quality of the products, like the higher yields, improving the quality of the product, change in the ploidy level of the plants, pigmentation for the ornamental plants which can be of the commercial importance, and so on. In fact, several somaclonal cultivars have also been released for the commercial cultivation. Depending upon the sources of the variation from where they originate, these are grouped in two categories. One is the pre-existing variability, and second is the in vitro induced variability. So the first category is the pre-existing variability. The variations that are already present in the explant when we harvest the explants to initiate the in vitro cultures can be categorized as pre-existing variability. In plants, somatic tissues accumulate mutations with the age and differentiation status. More is specialized is the tissue in the plant body, more will be the chances of the mutations present in those tissues. Further, more specialization means these cells are highly differentiated to perform the specialized functions. For example, the leaf versus meristem tissue. Meristem cells are continuously dividing and produce various organs of the plant body. Cells in the meristem divide by normal mitotic divisions, and usually uniform genomes are maintained in these tissues. However, the leaf cells are more specialized to perform photosynthesis associated specific functions. And during the process of differentiation for specializing functions, these cells may accumulate more variations. It means highly differentiated tissues have more variations as compared to the less differentiated tissues. The number and amount of variations increases with the age of tissues. That means a leaf tissue that is highly specialized would have less mutations when it is younger as compared to the mature leaf tissue. Even the duplication and endoreduplication may occur in the specialized somatic cells. That means the specialized somatic cells in a diploid plant may have different ploidy levels. In other words, aneosomatic or polysomatic conditions may arise in the somatic cells. And these conditions are very common in the vegetative parts of the plants. Therefore, chances of having the pre-existing genetic variations in the highly differentiated tissue is higher, and therefore extent of pre-existing variability that is contributed by the explant varies with the source of the tissue or the age of the tissue and health of the explant too. So let's learn a little bit about the polysomatic. Now the polysomatic is the condition where different cells of an organ or different tissues in the plant body may have different ploidy levels, and this condition is called as polysomatic. One of the major reasons for the polysomatic is endoreduplication of the genetic material in the cell. The term endoreduplication represents the condition where DNA synthesis cycle takes place within the cell. However, it is not followed by cytokinesis, that is the cell division. The DNA duplication may occur numerous times, and therefore the ploidy level in the different cells may vary significantly. Endoreduplication is actually an evolutionary strategy to amplify the desirable genes that are required to synthesize the necessary products for the plant growth. It has been suggested to enhance the physiological functions of the cells, as the larger volume will help in tissue expansion and would also help to raise the copy number of the functional genes within those cells where endoreduplication occurs. Endoreduplication usually results in increase in the size of the tissues or the organs. However, the signals that are necessary to change the classical cell cycle to the endoreduplication are still not clear. Endoreduplication events are more common during the cell differentiation process or the cell expansion, and specifically its occurrence is higher in highly specialized cells. Though it is still not clear that if the endoreduplication is genetically programmed or it is just the consequence of the differentiation and environmental stimulation. But certainly various environmental factors like light, temperature, nutritional regimes, or the application of various growth hormones directly influence the degree of endoreduplication. So the overall polyploidization of the somatic tissues may contribute to the somatic or the somaclonal variations significantly if the explants are prepared from these tissues having polyploidy or polysomatic. Second source of these variations is the in vitro induced variations. By now, we have understood the in vitro regeneration process in previous lectures. We select the donor plant and suitable explant to initiate the cultures. The explant is excised from the parent plants and incubated on the suitable nutrient media in the aseptic cultures or aseptic conditions. Any of the genetic variations that are present in the explant tissues when we harvest it from the donor plant is called as pre-existing variability, as we discussed in the last slide. And any variations that will be caused or introduced during the culture now on are called as the in vitro induced variations. So the explant tissues undergo various processes of regeneration like dedifferentiation, redifferentiation to generate the different organs and other morphogenetic responses. Basically, the differentiated cells in the explants are induced to dedifferentiate to make the callus cell. Callus is the mass of undifferentiated cells. Once the enough amount of callus tissue will be formed, these are induced for morphogenetic responses, that is the organogenesis. Then the shoots of suitable size are excised and transferred onto the rooting medium. After the roots develop from these shoots, the plantlets will be ready for their transfer to the soil for hardening. This whole process of in vitro regeneration could be very stressful or a traumatic shock for the living tissues and may result or instigate highly mutagenic processes leading to the mutations or abnormalities in the in vitro regenerated plants. Like when the explant is excised from the parent plant or the donor plant and transferred onto the in vitro culture media, it causes the wounding response, which may be followed up by the sterilization process, which is also stressful for the plant and harsh. Then the induction on the artificial media for dedifferentiation, followed by redifferentiation, or the excision of the shoots and transfer to the rooting media where they are induced for the production of roots. Now all these processes or the changes are very stressful for the living tissues and can lead to the oxidative stress on the growing tissues, which can result in the introduction of genetic mutations. Growth regulators can have significant impact on the introduction of somaclonal variations. The potential of the growth hormones or growth regulators may vary among various growth hormones, and also it can vary among the different plant species. Now, what are the various factors that affect these in vitro induced variations? There may be actually several factors that can result in the accumulation of in vitro induced variations. Majority of these can be categorized in the following groups, that is age of the culture, composition of the culture media, or the growth habit of the tissue or the inherent sensitivity of the donor plant genotype. The first one is the age of the culture. In the in vitro cultures, tissue is continuously induced for the multiplication at a high rate and it can affect the genetic stability of the tissues. Generally, the frequency or the rate of the accumulation of variations increases with the increase in the number of multiplication cycles. Further, the time period for which the culture is maintained under in vitro conditions is critical, and longer duration of the culture maintenance increases the frequency of somaclonal variations. Then second is the composition of the culture medium. Now, during the regeneration process, right from the explant inoculation or incubation on the culture media to the regeneration of the plantlets, at every stage of the tissue culture, different plant hormones are used. These may be used in the different combinations or at different concentrations. There are several hormones that are used in this in vitro regeneration process, and they are usually grouped in auxins and cytokinin categories, which are most frequently used. And use of these growth hormones is also associated with the accumulation of the somaclonal variations. There are several auxins like naphthalene acetic acid, indole acetic acid, indole butyric acid, or 2,4-D. And similarly, several cytokinins are there like kinetin, vinylaminopurine, or zeatin. And the potency of these hormones to cause the somaclonal variations differs significantly among different hormones. Like 2,4-D has been found to be the most potent for introduction of these somatic variations. Either it induces the polyploidy or selectively favors the division of the polyploid cells. In general, the in vitro morphogenetic responses seems to be dependent on the use of the plant growth hormones or the media composition. However, the genotype of the parent plant also has a significant impact. The regeneration capacity of the plants is regulated at the genetic level. Therefore, the nature of the genotype and their growth habits can influence the frequency of the regeneration, and hence it directly affects the frequency or the capacity for the production of the somaclones too. So what could be the mechanism of these somatic variations which can result in the somaclonal variants? There may be numerous mechanisms that can result in the accumulation of these somatic variations. However, some of the major categories where evidences have been recorded includes changes in the chromosome number or the structure, then smaller gene mutations, DNA amplification, methylation status of the DNA, or the activation of the transposable elements. So the first one is the change in the chromosome number or the structure. Change in the ploidy level are commonly observed in the tissue culture raised plants. These may be the result of the pre-existing variability. That means the ploidy level changes were already there in the cells of the explants when these explants were harvested from the mother plant and were introduced in the in vitro cultures. And these changes might have resulted because of the endo-reduplication as we discussed earlier. However, it may be introduced or amplified in the in vitro cultures too, like polyploidy is favored or preferred in the in vitro cultures. For example, if we mix the diploid and tetraploid cells of the carrot, when we grow these diploid or tetraploid carrot cultures individually or separately, the growth rates of these cultures are very similar. Now, if we mix them in the equal ratios and grow them together, the frequency of the tetraploid cells slowly increases in the culture, and after some time, finally, the whole culture may become a tetraploid culture. So, polyploidy is usually favored in the in vitro cultures. In vitro culturing have shown to introduce the structural changes in the chromosome too. These may include like the deletions, that is the loss of genes or the DNA segments, inversions, that is the alteration of the order of the genes on the chromosomes, or the translocations, that is the movement of the DNA segments from one chromosome to other chromosomes. Next is the gene mutations and DNA amplification. Chromosomal level changes occur at the large scale where larger fragments or the segments of the genome are involved in the variations. However, stress in the in vitro cultures may result in the introduction of small specific mutations, and the multiple mutations can accumulate during the process of regeneration. These may result from the culture conditions or the accumulation of the metabolic product itself in the media may have the mutagenic effects. The introduction of the somaclonal variations may not be totally a random process. Some part of the genome may be more susceptible for the variations as compared to the other part of the genome, and hence chances to interfere with the traits that are regulated from the susceptible part of the genome would be higher. However, these mutations cannot be directed or controlled to the specific part of the genome. Sometimes the changes may be on the recessive alleles, and hence these may not be expressed in the first progeny of regenerated plants. However, these variations may appear in the later generations or in the self-progenies. Further, the amplification of the specific DNA segments have been reported in many somaclones. In fact, it has also been associated with the new traits or the novel traits in the somaclonal variation based new cultivars, like the amplification of the glutamine synthase gene in alpha-alpha. Several fold amplification of this gene encoding for glutamine synthase in the alpha-alpha is directly correlated with the many-fold enhancement of the herbicide resistance in the somaclonal variants. Next is the methylation status of the genetic material. In many cases, abnormal plants that are obtained in the tissue cultures, they revert back to the normal phenotypes in the next generation. And usually these kind of changes which revert back, they are caused by the epigenetic regulations. In vitro cultures can directly influence the methylation patterns in the regeneration plant, and the hypomethylation patterns are more commonly recorded in the tissue culture raised plants. Growth regulators have a significant effect on the methylation patterns of the plant. Different growth regulators may induce specific type of methylation, like use of kinetin for the induction of the morphogenetic responses has been shown to cause extensive hypomethylation of the DNA, whereas the oxynaphthalene acetic acid has been shown to cause the hypermethylation. Similarly, other growth regulators may have a different kind of impact on the methylation pattern of the genetic material. However, these epigenetic changes are usually reversible. Then the next is the transposable element activation. Transposable elements are the mobile DNA segments that are capable of moving from one position to another position in the genome and can result in the insertional mutagenesis. It has been observed that the activity of the transposable elements usually increases in the in vitro cultures in the plants. So the tissue culture stress can activate the transposable elements that were silent earlier, and this may result in the accumulation of the genetic variations in the somaclones because of their activity. Next is the methods or the isolation procedures for the somaclones. Now the procedures for generating the somaclones is similar as establishing to the in vitro cultures. However, depending upon whether the selection is used during the in vitro culture or not, these can be grouped into two categories. First is the in vivo selection, where the somaclones are generated without the use of any selection in the in vitro culture media. And second is the in vitro selection, that is, selection agents are usually applied during the in vitro cultures. First category is the in vivo selection. That means in vitro culture is performed without any selections. Explants are harvested from the donor plant and inoculated on a suitable culture media in the aseptic condition. Regular tissue culture procedures are followed and the plantlets are regenerated. That is, these cultures or cells are induced to differentiate, which result in the callus formation. Once the enough amount of callus have been formed, it is transferred onto the regeneration medium, where the calli are induced for organogenic responses and shoots are regenerated. These regenerated shoots, when they grow to the suitable size, they are transferred onto the rooting media, where the shoots develop roots, and now the plantlets will be ready for their transfer to the soil for hardening. In this way, large number of plants are regenerated. Now these plants are ready for the screening of the desired trait. Suppose the desired trait is resistance to a particular pathogen infection, so these plants or the field adapted plants are infected with the pathogen and screened for the identification of resistant plants. Now, because of the infection, all the susceptible plants will die. However, if there is any variation in any plant that has gained tolerance or the resistance to this pathogen, it will survive. Like here in this picture, blue color have been used to represent one resistant plant which survives from the pathogen infection. It means that particular plant that has accumulated the genetic variation that is providing the resistance against this pathogen. So finally, the selected plants are multiplied and they are again tested few more times for the susceptibility, and finally selected somaclonal variants having the desired traits are ready and they can be defined as a new cultivar and can be released for the commercial cultivation. Next is the in vitro selection. So basically in this case, the selection for the desired trait is performed in the in vitro culture stage. Like explants are cultured on the suitable culture media and induced to differentiate. Once the enough callus has formed, this callus may be fragmented into the several smaller pieces and cultured separately in the individual culture vessels. Basically, the callus proliferation and maintenance is performed. In this process, enough callus material will be generated and it can be used for the selection process. Therefore, these calli can be exposed to the selective agent, which may be an inhibitor or a toxin from a pathogen that causes the disease, or it may be simply a abiotic stress factor like the high salt concentration if we are looking to identify the somaclones that can grow in the salty water, or maybe the temperature sensitive high heat or the drought conditions. So these calli are cultured on the media supplemented with the selective agent. Now in this media, only those calli or those cells will survive that would have accumulated specific variations or mutations that provide protection against the effect of the selective agent. Like here, one of the plant, one of the calli survives the selection of the selective agent. These calli which survive on the selection are multiplied and screened for the resistance, and this cycle is repeated at least a few times. Finally, the resistant calli that are selected after few cycles of selection are transferred onto the regeneration media, where they are induced for the organogenetic responses. Once the shoots are regenerated, shoots of the sufficient size are transferred onto the rooting media, where they are induced for the root production and they form the plantlets. Finally, the regenerated somaclones are transferred to the soil for hardening, and again these plants are tested for the desired trait in the field condition, and this cycle of testing is repeated few times too. The major advantages of the selection during the in vitro culture is that millions of cells can be challenged with the desired stress like the fungal toxin or the salt concentration or the herbicide in a relatively very small space, that is the in vitro cultures itself. Further, all these procedures are performed under controlled conditions with the small inputs with respect to efforts as well as the resources. As the selection for the desired trait is made at the initial stages of development, it is less time consuming as compared to the in vivo approach when the selection is not used in the tissue cultures. So in that case, lot of effort and resources need to put in the regenerating a large number of somaclones for screening, most of which usually will not be useful. So in this way, somaclones are created and several traits have been introduced in the various agricultural crops and have been released for the commercial cultivation too. Now, how to detect the somaclonal variations? Once the somaclones are created, evaluation of the somaclonal variations for the early detection is essential. Early detection of the variation can result in huge cost savings, like efforts, resources, and overall productivity would also increase, which is a kind of the basic requirement for commercial success. Based upon the type of the trait scored and the methods used to score these variations, these can be grouped into 4 categories. That is morphological traits, physiological traits, cytological methods, or finally the DNA or protein-based molecular markers. First is the morphological traits. It is one of the oldest, easiest, and extensively used method to detect the somaclonal variants. No special equipment is required and the variants can be detected with naked eye. Various morphological traits that have been used in the past for variant screening includes morphological traits related to the leaves or flowers like size of the leaves or the flowers, their shapes or the colors specifically are associated with the flowers, or their number, type of the pigmentation that can result in different colors, plant stature or the architecture of the plant, and so on. But the disadvantage of the morphological traits is that only a limited number of morphological traits are available for analysis. In addition, these traits are strongly influenced by the environmental factors too, and traits selected may not be actually a somaclonal variant. Further, if the variations are caused by the epigenetic changes, these may be temporary and will disappear by the next generation. Next is physiological or biochemical markers. These are comparatively faster as these can be performed at the early stages of the plant development, like the light or temperature responses. The variants may be exposed to various levels of light and temperature regimes and may help in identification of the light or temperature responsive variants. Similarly, the tissues of the variants can be analyzed for quantitative and qualitative analysis of pigments like chlorophyll, carotenoids, or anthocyanins, and so on. However, good amount of expertise would be required to perform these type of biochemical analysis, and again the number of such traits or the abundance of such traits is also very limited. Next is cytological methods. We can also use the cytological methods to score the somaclonal variants. If the genetic changes are large enough that these can result in the chromosomal variations or structural changes, these can be detected by the karyotypical studies and the observation of the chromosomal aberrations under the microscope. But these are time consuming. Suitable expertise is required and it especially becomes difficult if the chromosome number of the species in question is large. Flow cytometry can be advantageous for this purpose as the large-scale ploidy studies can be performed easily. And even a very small modification in the chromosome number can be recorded using the flow cytometry methods. Next is the molecular markers. These are one of the most widely used markers for characterizing any kind of variants. These are either protein-based markers or the DNA-based markers. Protein-based markers like the isozyme analysis, as we discussed earlier also for characterization of the somatic hybrids. Isozymes are multiple molecular forms of the same enzyme and can execute the same function. Usually the electrophoretic bending patterns are studied and compared with the control groups, Three types of differences or changes are usually observed in these electrophoretic banding patterns. Number one is the altered electrophoretic mobility. That is the change in the mobility of any particular protein or the enzyme is recorded. Second is the altered level of the particular protein. That is the change in the expression level or the accumulation of particular protein. It may increase or decrease in the variants. And third is the loss or gain of the specific protein. So in addition to change in the expression level, its expression may be completely lost from the particular tissue, or it may start expressing in a particular tissue where normally it does not express. These markers though are very useful, however these markers are also limited by their number. And finally is the DNA based markers. The use of DNA based marker is most popular as it can generate important genetic information and their abundance is really high. These are most advantageous as these can be used at a very early stages of the development when the plant is still in the culture media. The DNA can be extracted from any of the plant tissues and can be used for analysis. Further, many of these markers are codominant in nature and are phenotypically neutral. Therefore, these markers are very useful to study the variations. Several different kind of markers or the DNA markers are available. In the earlier times, RFLPs, that the restriction fragment length polymorphism, or randomly amplified polymorphic DNA markers, or amplified fragment length polymorphism markers, or the analysis of microsatellites were more popular. However, the improvement in the DNA sequencing technology, availability of the rapid DNA sequencing technologies, and because of the sharp drop in the sequencing cost over the last few years, sequencing based analysis are outperforming all other type of markers and they are more popular now. However, PCR based markers are still popular and can be used to score the variants easily. So in this way, these are some of the important tools that can be used for the characterization of the somaclones. Next is the advantages of the somaclonal variations. Somaclonal variation is a tool to create new variants. Novel traits can be generated through this technique too. Variation can be introduced both at the nuclear DNA level or in the cytoplasmic genomes, that is mitochondrial or the plastid genome. So it means that the variation can be generated in the cytoplasmic traits as well as the nuclear encoded traits. These procedures are easy to perform and they do not require the sophisticated technologies or equipments to set up these facilities. Just the in vitro regeneration facility is required. Many novel variations that result in the novel traits can be achieved through the somaclonal variants, which cannot be achieved through the conventional breeding, and therefore these methods have a huge potential. Large population of cells, in fact millions of cells, can be used for the in vitro selection, and therefore when a large number of cells are exposed to the selective agents like the toxin or the salt or herbicides, chances of the success for identifying the somaclonal variants is also better. In vitro selections can further shorten the time period required to isolate the somaclones with the desired traits. New cultivars generated in this way would not have any socio-ethical hurdles, which is a major problem for the commercialization of the genetically modified traits. So overall, these somaclonal variations can be very useful and can be used to improve either the existing traits or even can be used to introduce the new traits or the novel traits that did not exist earlier in the plant species. Though it is very promising tool for the crop improvement, however, there are several challenges that we need to take care. Like the presence of these variations is undesirable if the aim of the experiment or the aim of the in vitro culture is micropropagation, as it may compromise the important traits like the yield or the quality compromise of the product in the elite cultivars, which is not desirable in these multiplications. Because the variation introduced are random and there is no way variation can be directed to a particular location of the genome, therefore large number of progenies needs to be screened to identify the desired plants. So it becomes labor intensive too. Most of the somaclonal variations that appear in the regenerated plants may not be useful because of their random nature and may result in the waste of time and resources. In order to work with the somaclonal variations based plant improvement, availability of an optimized regeneration protocol is a must. And if the regeneration protocol for the species in question is not available, it needs to be established, which is time consuming as well as the resource and labor intensive. The variations that are introduced through the in vitro cultures are unpredictable and uncontrolled. There is no way that these variations can be controlled or directed or restricted to a particular region of the genome. These variations may not be useful for the improvement of the complex agronomic traits. Further, in addition to developing the novel or desired traits, it may introduce the negative mutations in the other traits and compromises some other traits like the yield or the quality of the products and would not be desirable. Next is the applications. Since their characterization for the first time in 1970s, somaclonal variations have been used for improvement of several traits in the agricultural crops. It can be regarded as actually an efficient tool for breeders to generate the new cultivars. Several disease-resistant cultivars have been generated and commercialized, like downy mildew resistance in barley, resistance to fusarium species in several plants like banana, tomato, or celery, or the resistance to other diseases in sugarcane or brassica or rice, and so on. Similarly, the tolerance to the abiotic stress factor have also been introduced in some crops like the salt or heat tolerance in the flax. The crops with the resistance to the herbicides and insect damage has also been generated. Similarly, several other agronomically important traits like improving the quality of the product, better yield, fruit quality, or the male sterility have been introduced in several plants. Several other characters like thornless blackberries improving the cooking quality of the grains, or improving the nutrition quality or the yield in wheat and mustard, reducing the browning of the tubers in potato. These all traits has been introduced in these species through the somaclonal variations. The traits associated with the leaf and flower morphologies and different color variants have been introduced in some ornamental plants, and they are very useful for the flower industry. So overall, somaclonal variations have proved to be a good source of variation, which is useful for modifying the existing characters or even introducing novel traits that never existed in those plants. Indeed, several improved cultivars of important crops like sugarcane, barley, tomato, potato, banana have been released for the commercial cultivation. So in summary, we talked about the somaclonal variations. We learned about the sources of the variation, where we discussed the pre-existing variability or the in vitro induced variations. Then we talked about the molecular mechanism that causes these variations, like changes in the chromosome number and structure, gene mutations, DNA amplifications, or the epigenetic changes. Finally, we talked about the various methods used to generate the somaclones, followed by their advantages or disadvantages or various applications for the crop improvement. Thank you.